Welcome to session 3 of today's meeting. I'm excited to have 5 wonderful events this afternoon. My name is Michael Röhrl. I'm director of the Center for Precision Pathology at Memorial Sloan Kettering Cancer Center. And I've known Paul Hoffman for many years. So thank you for asking me to chair today's session. So I'm delighted to introduce the first speaker, Humam Kadara, from MD Anderson Cancer Center, where he's an associate professor in the Department of Translational Molecular Pathology. And the title of Humam's talk will be Field of Carcinogenesis and Phenotypic Evolution of Lung Cancer. Welcome, Humam.
Hello, can you hear me? Thank you for inviting me to present today on our work on field carcinogenesis and the phenotypic evolution of lung cancer. I was just promoted as a panelist now, so I'm just going to share my screen as we speak. So just be patient with me. Can you see my screen?
I can, yes.
Yeah. So thank you for the invitation to talk about field of carcinogenesis and phenotypic evolution of lung cancer. My lab, along with a group of wonderful collaborators, have been studying a phenomenon in the lung called the field of injury, where the respiratory epithelium can be mutagenized by exposure to tobacco or other inflammatory insults, and this leads to sometimes widespread molecular or inflammatory changes, these are depicted on the left, or these changes can be more localized in normal epithelium that is nearby the tumor, and this is sometimes referred to as field of cancerization. This principle, i.e. field of injury or field of cancerization, we and others believe is very important to understand how normal epithelium can be primed by being a field of injury to progress to preneoplastic lesions and invasive carcinomas. And this primed field of injury, we and others have shown to comprise molecular changes in the form of somatic mutations in key driver oncogenes and tumor suppressors, as well as copy number alterations. And we've published a number of publications highlighting these. And one of the recent studies we found was we characterized the genomic landscape of somatic mutations and copy number alterations in the normal appearing epithelium in folks with early stage adenocarcinoma or squamous cell carcinoma. Mostly they were adenocarcinoma, early stage adenocarcinoma patients. We cataloged events in the form of either point mutations or different types of allelic imbalance in the form of copy number gain or loss, or even copy neutral loss of heterozygosity. And some of these events were quite relatively common, such as, for instance, p53 mutations or Notch1 allelic imbalance. It is interesting to note that Notch1 alterations in the form of mutations or copy number alterations have been shown to be found in recent publications in the normal appearing epithelium of folks without cancer, such as those that are undergoing aging. The Notch1 alterations have been found in folks in the normal appearing epithelium in the esophagus, skin, even other squamous type tissues that are normal, and in people who are cancer free and phenotypically healthy. Previously, when we first reported these findings, people were skeptical on the amount or frequency of genomic alterations we're finding normal epithelium, but with the advent and of these different studies characterizing the genomic landscape of alterations in normal epithelium of folks without cancer, there has been a rising interest in understanding bottleneck evolutionary constraints in normal epithelium. Along on that theme, we've also been studying normal lung tissues, preneoplastic lesions, and matching lung adenocarcinomas, mostly from mostly lesions in the sequence of pathogenesis of lung adenocarcinoma, where we have documented by a variety of techniques, including deep DNA sequencing and genome-wide array profiling, as well as RNA sequencing. We've documented changes that are progressive along the pathologic continuum of normal lung tissues, atypical adenomatous hyperplasia, and lung adenocarcinoma. And we were among the first groups that found a decrement of immunity along this pathologic continuum, with many of these immunological or immune-centric gene expression programs changing beginning at the preneoplastic stage. We even found that by deconvolution analysis of these immune-centric gene expression programs, that there is a decrement in either an upregulation of CD4 positive T cells or downregulation of cytotoxic CD8 positive T cells as we go from normal lung tissues to early lung adenocarcinoma precursors to lung adenocarcinoma. Most of these studies have really centered on bulk sequencing analysis of lung carcinogenesis. The advent of single cell technology has really shed light on the roles of different cell populations that may play important roles in not only cancer progression and metastasis, but also response to therapy. However, the roles of these individual cell populations in the spatiotemporal evolution of lung adenocarcinoma, meaning from by the acquisition of a mutagenized normal appearing field or the transition through preneoplastic lesions, is very, very poorly understood. And so we really asked the question what the cell is going on, and undertook a effort to understand the single cell landscape of early stage lung adenocarcinoma development. And so, based on our interest in the field carcinogenesis phenomenon, where we believe that the adjacent or very nearby epithelium may inform of the development of the tumor that is just adjacent to this normal epithelium, we believe that there's this geospatial heterogeneity in normal appearing tissues in the lung, from which the lung tumor arise. We performed the study design in which we performed single cell sequencing of lung adenocarcinomas, as well as multiple spatially distributed normal lung tissues, those that are adjacent, intermediate, and distant relative in spatial proximity from the primary tumor. We have defined the spatial distances of these samples to range from 0.5 centimeters from the primary tumor, the adjacent normal, to up to 9 to 11 centimeters from the primary tumor, which is a distant normal. And one of the first analyses we've done was to really perform an unbiased analysis of the cellular ecosystem of lung adenocarcinoma and these normal appearing lung tissues. And we found that the fraction of epithelial cells is quite little. It's around 3.5 to 4%, 5%. And this is in line with recent studies of solid tumors, because we're very much interested in the field carcinogenesis phenomenon, and we want equal representation of different populations. So we did a study design where we enriched for epithelial and non-epithelial cells by sorting based on EpCAM expression, and we generated libraries from these enriched cell populations. And in this way, we did this for a number of patients, and we were able to sequence quite a relatively large number of cells with a good representation of epithelial cells and a good representation of the different major lineages, such as myeloid, lymphoid, endothelial, stromal, and epithelial cells. Here we jumped from a 4.2 to around 38%, and we're even able to capture rare populations such as proliferating sublineages. So using this unique cohort of a single cell cohort from early stage lung adenocarcinomas, we looked at the variation of these different cell populations in space in space means macro space, from distant normal lung tissue to the primary lung cancer. And we found a variety of different cell populations that change geospatially. They either decrease gradually or increase gradually, or they're depleted sharply in the tumors, such as natural killer cells, or are massively increased in the primary tumors compared to the uninvolved normal lung tissues, such as B cells, which I will come back to later. This work has been recently published, actually just out this month, in Cancer Discovery. To keep the story brief, we're going to show you a couple of vignettes that came out of our analysis. This is a Humam projection of the different lymphoid populations that we found. We were able to identify a number of different CD8 and CD4